What are sense organs? Sense organs are the organs in our body that help us detect stimulus from our environment. For example, vision, hearing, sense of smell, taste, and touch. All these are our senses, and the organs associated with them are our sense organs. Other than that, we can also sense the equilibrium of our body. We can sense how we balance our body. We can sense changes in temperature, pain, and pressure. Proprioception is our capability of sensing how our body is moving in a three-dimensional space. So there are different kinds of receptors in our body that make up these sense organs. For example, mechanoreceptors that feel the mechanical pressure, vibration, touch. For example, our skin can feel that, our ears can feel vibration. Thermoreceptors are the receptors that can feel temperature changes, and photoreceptors are those receptors that can sense the changes in light. Chemoreceptors detect chemical signals. Such chemical signals are processed by our nose as well as our tongue, and nociceptors are the pain receptors. Let's talk about the sense of vision. The organ responsible for the sense of vision is our eye. The anatomy of eye shows us that there are three main layers called sclera, choroid, and retina, starting from outside to inward. When this sclera comes in front of our lens, which is here, it becomes cornea. Then the entry of our light into our eye is regulated by iris, which is this structure, and the pupil, which is the opening of the iris. So the light enters our eye through pupil, and this light is then converged by the lens and focused on the retina at fovea centralis. So fovea centralis is the main area where we perceive light. Retina has two main kinds of cells called the rod cells and cone cells. Rod cells help in detecting the intensity of light, and cone cells help us in determining the color. All the information from the photoreceptors present in retina of our eye is converged by coming together of axons. So all over the retina there are several neurons, and those neurons have their axons, which when converge at one point end up making the optic nerve. So let's see how the visual information is processed in our brain. The light enters through the lens and it is converged at the retina. So this is the outermost to the bottommost arrangement of cells in the retina. So the light goes all the way down and is received at the photoreceptors present at the base of retina. Here some chemical reactions happen, and the pigments in our photoreceptors get excited and they pass the nerve impulse down the axons of these photoreceptor cells. Now you can see there are synapses and several other kinds of neurons which are making junctions and synapses, and finally this information reaches the ganglion cells. The ganglion cell axons then converge and bundle up to form the optic nerve. This optic nerve is basically a cranial nerve, which is the nerve that arises from the brain, and optic nerve takes the information of visual signal to the visual center of our brain. And where is this visual center located? It is located at the occipital lobe of our forebrain. So as mentioned earlier, the retina, okay, so this piece of retina has been zoomed in. So you can see that the outer layer is this and the bottom layer is this. So photoreceptor cells are basically at the base of retina. So here you can see two kinds of cells. The one cells which kind of looks like a rod and the one which looks like a cone. So these are rod and cone cells of retina. Both these cells are photoreceptor cells. The rod cells help in day and light vision. That means it tells you about the contrast of light, and cone cells help you understand color. The rod cells are present at the peripheral region of the retina, and the cone cells are present at the fovea. So, fovea, which is also called the fovea centralis. Rod cells are very sensitive. Even slightest change in the intensity of light, even if your eyelids are closed, you can still feel that when you cover your eyes, you would feel that there is darkness, and if there is bright light outside, even with your eyes closed, you can feel that the intensity of light is changing because the rod cells are very sensitive. And rod cells therefore help in the black and white contrasts, and cone cells help in color vision, help us understand basic colors like red, green, and blue. Some common disorders related to eye are myopia, which is nearsightedness, when a person is able to see the near objects clearly but can't see the distant objects that clearly. Hyperopia or farsightedness is when someone can see the far objects clearly but not the near objects as clearly. Astigmatism is when there is blurriness in the vision, mainly associated with the irregular surface of our cornea, and the light is bouncing off in all the directions and it's not being able to focus at one point. Glaucoma is a condition in which there is so much pressure built inside the eye because of the fluids present in the eye that it ends up damaging the optic nerve. And cataract is the cloudiness of lens. So the lens loses its transparency and becomes more opaque. Next, we'll talk about the sense of hearing and balance. The organ associated with hearing and balance is our ear. So this is the outer part of the ear, which is shaped in a way that it gathers all the vibrations from the surroundings and converts them into this tube, which is called the external auditory canal or the ear canal. The vibrations are taken to the eardrum or the tympanic membrane, the vibration of which will then be transmitted to the middle ear. The middle ear has 3 ear ossicles or ear bones called malleus, incus, and stapes, and they enhance the intensity of sound or amplify the sound, and these vibrations are then taken to the inner ear. And inner ear has this structure which is divided into 2 parts. The upper part is called the semicircular canal and the lower part is called the cochlea. Out of these, cochlea is associated with understanding sounds and hearing, and semicircular canals are associated with detecting balance, posture, and movement. So how do we analyze the auditory information? The vibrations gathered by the external ear are taken to the eardrum. The vibration of eardrum is passed on to the ear ossicles, which then take these vibration to the cochlea. Inside this whole structure called the labyrinth, there is a fluid circulating, and the vibration that has been carried by these ear ossicles then starts vibrating the fluid inside the cochlea. The movement of this fluid moves the hair cells. So there are cells inside this cochlea which are neurons basically, and these neurons have tiny hair on their surface, or you can say that these are modified dendrites. So when the liquid around these cells start to vibrate because of the vibrations we have received from the sound around us, the hair starts to vibrate as well. So these vibrations are converted into electrical signal, and this electrical signal in the form of impulse is traveled down the axons, and these axons are bundled up together to form the cochlear nerve, and cochlear nerve then takes the signal to the brain, where it is processed at the auditory center of our brain. Along with cochlea, this structure has these three-dimensional arrangement of three semicircular canal placed in XYZ axis. Inside them also we have these specialized sensory cells. The movement of fluid or the movement of inertia causes these hair on these specialized sensory cells to move, and this movement of hair on the sensory cells is conducted into electrical impulse, and again that information is converged through all the axons and then taken to the brain through a nerve called the vestibular nerve. This three-dimensional arrangement of semicircular canal helps us understand our posture, our orientation in the three-dimensional space, and rotational or linear movement. Common disorders associated with hearing are loss of hearing, which can be caused by some problem in the outer or middle ear, which we can say are the problem of conducting the vibrations, or our sensory part of hearing is damaged, which is caused by the damage in the inner ear. The other kind of disorder is tinnitus, in which a person continuously hear ringing in their ear, and that is associated with damage of ear nerves. Vertigo is a situation where a person feels dizziness because of the inner ear dysfunction. So our inner ear is not able to process the information of orientation and height, and that's why a person starts to feel dizzy because they're not able to process that information properly. Next, we talk about the sense of smell. The organ associated with our olfaction or sense of smell is our nose. At the upper area of our nose, we have specialized olfactory nerves or olfactory neurons that gather or receive the chemical signal. All this information is then converged at the olfactory bulb, from which the olfactory tract or olfactory nerve is arising. This information is then taken to the olfactory center of our brain, and it works very closely with our sense of taste, because our sense of taste is also dependent on receiving chemical signals, and that information is processed to understand what kind of smell or taste we are perceiving. How does the sense of smell work? So the chemical signals from the source are dissolved in the ear, and then they are taken into the nose when we breathe. The olfactory neurons present in our nose receive this information, and this information is then taken to the olfactory bulb, from which the olfactory nerve arises, and it takes the information to our limbic system. Our limbic system then understands this smell, and we are able to relate it with our memory. We can think what kind of smell this is. Does it smell of some familiar food, or does it trigger some memory? And that information is basically obtained out of experience. Under normal circumstances, a person should be able to perceive different kinds of odor, but if they are perceiving one or more odor less than the normal, then they are said to be hyposmic. If they are not perceiving one or more odors completely, like the level is not low but it's completely gone, the sensation is completely gone, then a person is called anosmic. And if they have altered sense of smell, they are called dysosmic, which means you smell something but you think that it's something else, or some bad smells start feeling good or different. Next, we'll talk about the sense of taste. The organ related to our sense of taste is our tongue, and the tongue has taste buds on them, and there are 5 different kinds of tastes that we can perceive, which is sweet, sour, salty, bitter, and umami. In our taste buds, we have the neurons present, and we receive the chemical signals And this whole taste and olfaction work in coordination for us to better understand what are we smelling and what are we tasting. So how does the sense of taste work? The molecules of taste dissolve in our saliva and then they bind with the receptors present on our tongue. So this information from the receptors is passed down the axons and is then taken to the brain, where the brain is able to process this information about the food, the smell and texture in the gustatory center or gustatory cortex of brain, which is related to the sense of taste. A complete loss of taste would be called ageusia. Having an altered perception of taste is dysgeusia, in which we taste something but we feel it's something else. And a reduction in the sense of smell would be called hypogeusia. Next, we'll talk about the sense of touch. There are several kinds of touch receptors present in different layers of our skin, starting from epidermis to dermis to hypodermis. In the epidermis, we have Merkel disc, which help us perceive what kind of texture or shapes we are touching. Meissner's corpuscles help us with finer touches, like the fine changes or perception of texture, and they are mainly present at the fingertips or our lips. Ruffini endings, which are present in again our dermis, help us with the feeling of stretch or warmth. And Pacinian corpuscles are present deep in the hypodermis, and of course, because they're so deep, they can be stimulated only when we are feeling some deep pressure. So how do we perceive touch? When our skin is touched or it receives a stimulus, the mechanoreceptors, which feel the mechanical stimulus of touch or pressure, they get activated and they send the signal to our brain, especially this region of the brain called the somatosensory cortex. Then somatosensory cortex produces a response by sending this information to the motor cortex, and then motor cortex send the information through motor neurons to the part of the body where you have to produce a response, and tells your organs to, for example, muscles to contract so that you can take your hand away from something that you're touching. The disorders associated with the sense of touch could be neuropathy, in which the certain area of your skin has damaged nerve endings and you feel numbness or pain, or hyperesthesia, in which you have very heightened sensation of touch, or hypoesthesia, in which you have reduced sensation to touch. So to summarize, today we learned about different kinds of stimulus like light, sound, chemical or pressure, and then we learned how eye processes visual signal, nose perceives the auditory signal and helps us understand sounds and balance. Nose and tongue receives chemical signals and help us perceive the taste and smell, and skin helps us with the mechanical signals, help us understand touch, pain, pressure, and all these senses work together for better survival of an organism. We understand our environment, we receive these sensations, we receive the information, and we can work on these sensations to increase our chances of survival.